Assalamu alaikum. Welcome to this special live stream. I am again and delighted here again to be speaking to Dr. Paul Nelson, who you all know, especially if you've been following the intelligent design discussion for the best part of the last 30 years, in fact. So welcome, Paul, back to the channel, and it's great to have you.
Thanks for having me.
So, Paul, we were going to do this two-part series on testing universal common descent. So today and then in 2 weeks' time, and you have a presentation you want to share. So I'll just share that and over to you.
Great. Well, I am delighted to be with you again. And I should indicate that I will supply all the reference materials for this presentation to Subor for distribution if anyone wants to contact him. You can also reach me personally via the Discovery Institute, and their URL is here on the title slide. So this presentation on testing universal common descent has two parts. In the first, which we'll look at today, we're going to examine the logical framework of the theory. And then in part two, on September 18, we will look in some detail at the molecular data. Although we will get our feet wet, so to speak, looking at some biological data today as well. So the scientific universe in which we find ourselves, and by we I mean globally the scientific community, and in particular biology, is dominated by the Darwinian view of universal common ancestry. So as you know, late in the origin, I believe this passage comes from chapter 14, the great summing up chapter of the book, and this is the first edition. Darwin says that the affinities of all beings of the same class have sometimes been represented by a great tree. He extends that beyond the class level to all living things have much in common. And then continues the argument saying, I should infer from analogy that probably all the organic beings which have ever lived on this earth have descended from some one primordial form. Well, we can take that geometry of relatedness from 1859 and find its corresponding content in 2021. So affinities we understand today mainly via molecular data, DNA, RNA, proteins, the ubiquitous molecular machine of the ribosome, which in part two I will discuss in significant detail. I think ribosomes are absolutely fascinating. A great tree, of course, is what we're calling universal common descent. And lastly, some one primordial form is today understood as the last universal common ancestor, abbreviated LUCA. So this topology of relatedness, or this geometry of relatedness, has had a pretty good run. 150 plus 160 years of dominating historical biology, and actually more than just the historical dimension of biology, it informs our understanding of comparative biology generally. When you look at gene sequences, typically a biologist will locate those somewhere within this great branching tree. So here is a standard textbook representation from Norm Pace, published in 2009, where we have the three major domains of life. The bacteria and the archaea are both single-celled organisms lacking nuclei. The eukarya is all those organisms with nuclei. We, of course, are on this branch here, the animals. And even though this doesn't look like a tree, one can imagine that it could be a great oak tree that you're looking at top down. In other words, you're hovering above the tree, looking down on it with its three major domains. And the last universal common ancestor then would be located here at the origin, although the last universal common ancestor cannot be identified with the origin of life. I won't have time to explore that today, but theoretically, one can argue that there is a distance between the origin of life itself and the last universal common ancestor. The key logical point about the did I just lose my screen?
Yeah, what happened is you, I think, clicked on another tab.
Oh, I meant that was accidental. Let me get the
Sure.
share back. I am very sorry about.
No worries at all. I mean, just to reiterate the previous point, which I think is important. Usually, it's almost seen as self-evident that the last universal common ancestor is the same as the origin of life. That there's just a link between the two, but there actually isn't. So we have Dr. Paul back with us.
I'm very sorry.
No worries.
I try not to touch, bump any buttons that I shouldn't.
There you go.
All right. So the key logical point about universal common descent is indeed its universality. All organisms on Earth share a common ancestor. So there's no organism outside that domain. And that is a very attractive feature to biologists, which I will talk more about in just a bit. So the question is, can this theory be tested? Now, perhaps counterintuitively, the short answer is no. And I'll go into the logic of that in just a moment. But the other short answer is yes. Now, you may say, well, look, Paul, that's a contradiction. Both of these answers cannot be the case. Either it can be tested or it can't be tested. Well, the point of my presentation today is to explain why this apparent contradiction exists in evolutionary theory and why it matters. How can we say yes and no to the question, can this theory be tested? And as I've said, this is important because universal common descent is today, in September 2021, the canonical historical geometry for life on Earth. It's what one learns even in primary school. And certainly if you pursue a graduate education in biology, this is the geometry that is taken as given, taken as the case. But as I'll explore in some detail in part 2, universal common descent is coming apart at its base. In other words, the tree is fracturing at its base. And that fracturing is extending up into the branches under increasing pressure from unexpected genetic data. And it's very important for us to understand the logic of this fracturing as biology goes forward into the 21st century. So here's a quick outline. We'll define universal common descent. Today, we will ask, how does it generate predictions? And then we'll give the no answer to whether it can be tested. In part 2, we'll look at what I call a complex bundle, borrowing a notion from Pierre Duhem and the great logician, the Harvard logician Quine. And we'll look at the yes answer. So we'll consider both of the answers and then point out why the question and the answers are important. So logical analysis today, although, as I said, we will dip our toe into some biological data. And then in part 2, we'll look at the genetic code, the ribosome, key essential genes and the major domains and so forth. And we'll look, I hope in not excruciating, but in some significant detail at molecular data that challenge universal common descent. So this will be our topic, or these will be our topics for today. And this matters. It does not serve us well to skip the logical framework because it's only within its structure that the data have their full significance. All right, so let's define universal common descent. Now, we should start with a widely held view that I encountered first as an undergraduate, but then throughout my graduate training and even today, where universal common descent is seen as a deduction from a pair of what are taken to be self-evident biological premises. So Keith Stewart Thompson, a biologist for whom I have a great deal of admiration, argued that it was in fact a deductive inference from these self-evident biological premises. And he put it this way. He said, the hypothesis of universal common descent is a very strong one based on a tight logic. It can be derived from the twin premises that life arose only once on Earth and that all life proceeds from existing life. Given these premises, and if the pattern of diversity among organisms has changed over time, then it must follow that any new forms have appeared as the result of a process of descent from pre-existing forms. This is based upon such unassailable logic that we act as though it is a fact. And this paper was published in the early 1980s. And indeed, some biologists, surprisingly many in my estimation, have argued that universal common descent is actually an axiom or first principle of biology. So Kenneth Weiss at Penn State, in an article in 2001 entitled "We Hold These Truths to be Self-Evident," he argued that among the basic principles of Darwinian biology, and this lowercase d is not a typo, it's his estimation that Darwinian has now become so widely accepted that we can treat it as a generic modifier, adjective that does not need to be capitalized. Those principles are, one, that all life came from a point source, and secondly, that the assumption of the point source is so important to the entire theory that we insist on it at the beginning, but forbid it at any other times. And he says, is there anything else in biology that's as inviolate, in other words, that we take as a given, like shared ancestry? Richard Lewontin died last month, or actually, in July, on July 4. One of my scientific heroes, had a long career at Harvard. Prior to that, at University of Chicago, he actually was a mentor to my PhD advisor, Bill Wimsatt. And in a letter in 1994, he said to me, Paul, let me give you the materialist understanding of evolution, by which he meant universal common descent. And he put it this way. He said, let me sum up the materialist position. We take it as given that one, living material arose from non-living only once in the very distant past. And for various reasons, those organisms had to be what we now call simple. They could not have been a metazoan, that is an animal, or a vascular plant. He went on, since that time, because of the nature of living organisms, which tend to eat everything in sight, no further arising of life from non-life could work. And therefore, since that time, we have the law of all life from life. This is known, by the way, more formally as the law of biogenesis, which Peter Medawar said is the most important principle in biology. Medawar actually argued that evolution itself could be seen as an implication of the law of biogenesis. So you see the correspondence here between Lewontin's view and the view of Keith Stewart Thompson that I discussed a moment ago. And then he said, 3, there are organisms on Earth today that were not here a couple of billion years ago. They're not here anymore. He said, if you take these principles as given, evolution follows as night the day. That is, the kinds of organisms that are here now didn't used to be here, and they had to come from simpler forms. That's what we mean by evolution. All the rest is commentary. He acknowledges that these are postulates that he cannot prove, but he takes as self-evident. And there's a certain elegance to this deductive formulation. If life did begin with a single event of abiogenesis, that is the origin of life from non-life, after which that process ceased for whatever reason, then if the law of biogenesis holds, omne vivum ex vivo is the Latin tag, all organisms stem from other organisms, then life, no matter how diverse, blue whale, oak tree, starfish, doesn't matter. It must trace its ancestry to that abiogenesis singularity. So this would be the realm or the domain of universal common descent. And these authors and others treat this as an axiom or first principle. And prima facie, it appears sound, even elegant, this formulation, this deductive formulation. The problem is this formulation renders universal common descent untestable. And this isn't something that one needs to do any observation about. You can grasp the problem from your armchair. It's a logical conundrum. So here's our deductive formulation represented schematically. If we try to test it, it must be possible to observe at least one organism which did not stem from the abiogenesis singularity. It must be possible that there are, for testing, that there are organisms that do not fall within this domain. In other words, this domain would not be universal with respect to life on planet Earth. In other words, we have to be able to detect these other abiogenesis events if they occurred and left progeny in the biosphere. The problem is we've already stipulated that life arose only once, rendering these other events invisible. If the law of biogenesis holds, if we stipulate a single abiogenesis event, a singularity, we eliminate the possibility of detecting any other such event. And by stipulation, by the very structure of the deduction, no testing is possible. Okay? Now, there is a certain beauty, as I've argued, to this deductive formulation. You can obtain the truth of the theory with almost no effort. Just set up your premises the right way, turn the gears, and you get the universality of the domain of universal common descent, as I said, with basically no effort. The problem is there's a very high cost, and that is empirical content and testability are lost entirely. So universal common descent as a deductive inference fails if one is concerned to have the theory be potentially vulnerable to observational challenge. If you care about testing, in other words, this is a philosophical desideratum. If you care that your most general theory of biology is testable and not an axiom, then this is not going to work because there is no testing possible for this formulation. You're wrecked on a logical shoal. And I think when I've discussed this with biologists throughout my career and brought this to their attention, that they're holding the theory as an axiom, they don't like it because they recognize that science must have traffic in both directions between theory and data. That's got to be a two-way street. In other words, our theories give us expectations about the world, and then the world talks back to us. The data talk back. So if testing is something you want to hang on to, you cannot hold universal common descent as an axiom. It must be vulnerable to empirical challenge. So we're going to set aside these deductive formulations as fundamentally unsound with respect to our larger framework of wanting the theory to be empirically vulnerable. All right, so let's look at some standard textbook formulations. And these are textbooks that I used in my graduate training. Actually, this goes back to my first year as a biology student at University of Pittsburgh. I just pulled these off the shelves of my office here in Chicago. But you'd find pretty much the same formulation in any biology textbook. And you'll notice as you read through these that they're all expressing in different ways the same claim, namely this unified domain that terminates in a single ancestral organism. Here are another couple. This is from I believe this is Matt or Mark. I can't remember which Ridley this is. I'm sorry about that. But again, this is Peter Axe. These textbook formulations, in a variety of ways, you could boil these down to a single proposition, and that is all living things on Earth whether extant or extinct, trace their ancestry to Luca. Now, this is very, very important, this next point. Pay close attention to the nouns that are used in these formulations. Species, single common ancestral species, life, ancestor, organism, ancestor, descendant. These are generic common nouns. Let's look at the first three. Organisms, life, common ancestral cellular organism. These are at the most general end of the spectrum in terms of a biological descriptor. They're entirely generic. And we can list some of them here. They're general. These common nouns apply to any organism or species, and thus to no organism or species in particular. So our host, Zubor, is a living thing. I am a living thing. My cat, Newton, I can see from where I'm sitting over there in the corner of my office, he's a living thing. So these are the bacteria in my large intestine are living things. These are very general. And that's something that for a theory of the scope of common descent, you actually want. You want the theory to be formulated in the most general terms possible. So universal common descent gains its universality by abstracting away from the specific features of living things to the most general. And this is true as well for the process or mechanism terms that we have in textbook formulations of universal common descent. So let me just highlight those here. Again, notice their generality: linked, related, descended, existed. There's another related. A connects to B, right? We're not talking about sexual reproduction or asexual reproduction or any particular mechanism. It's just A is linked to B by an abstract relation of ancestor-descendant transmission, right? Descended, diversified, produced, connected, reproduction. So the verbs of action within the theory are, again, as general as they can be and still stay within biology. They describe abstract relations that, again, apply to any organism or species and thus to no organism or species in particular. Now, this perfect generality is very attractive to biologists as well as to other scientists for good reason. Scientists want theories that compress as much as possible into as little as possible. That's one of the functions. In fact, I would say it's perhaps the cardinal function of our most comprehensive theories, is that they abstract away from particulars and give us the most general propositions that hold as the case across the whole domain of experience. Therefore, not surprisingly, UCD is often expressed as a single general proposition, like this: all organisms on Earth have descended with modification from a unique cell, the last universal common ancestor. Okay, now, we're not going to hold this as an axiom. We've already seen that that won't work if we want the theory to be testable. So we need to know, what does this predict so that we can test it via observation. Now, here the problem takes a counterintuitive turn. And depending on how much you've thought about this, it may be more or less counterintuitive, but it's definitely surprising. So the easiest way to explore that counterintuitive turn will be a pair of biological thought experiments. So let's segue to part 2. How does universal common descent generate predictions? So we need to adopt a basic model of hypothesis and theory testing, and I'm going to use modus tollens. This is Philosophy of Science 101. We have some theory that predicts or entails a set of observations, and we just look to see, do those observations obtain? Is it the case? If they do, the theory is provisionally confirmed. We're good to go. Haven't falsified it. We can keep using it as our tool. What we see is the case, the observations that we predicted are there, consistent with the theory. What happens when they don't obtain? Well, the theory is potentially disconfirmed or falsified. Now, of course, in practice, hypothesis and theory testing is far messier than that. But we'll start with modus tollens before we get into the weeds. So we want to know, what does universal common descent predict that we can observe? Excuse me. So let's try our first thought experiment. And the point of this experiment, just to let you know where we're going, is we want to know whether in the case of some system, some biological system, do the observations confirm or disconfirm universal common descent. So keep that in mind. That's where we're going with this thought experiment. Is what we're going to describe, do those observations confirm or disconfirm UCD? All right, so there's our question. So we need a knowledge base to motivate the thought experiment. And if you've had some molecular biology or cell biology, this will be familiar to you. Here is a very cartoonish representation of the ribosome. By the way, when you see a figure like this in a textbook, where it looks like a big marshmallow or, piece of taffy, you can be sure that a great deal of detail has been omitted. So in this case, in defense of the artist, the detail has been omitted so that we can make out the key features of the ribosome that are being described. In this case, I want you to focus on the role of release factor, this protein here in the A site of the ribosome. So the ribosome is moving this way, 5' to 3', along a messenger RNA strand. And when the ribosome arrives at a stop codon, there is no transfer RNA that corresponds in the universal code to this signal, because it's the end of the reading frame, right? The protein here coming out of the tunnel in the large subunit has been assembled. It's still attached, however, to the penultimate the tRNA in the penultimate codon position is still attached here. This amino acid is still attached at the CCA stem of the tRNA. And if we can't find a way to cut the protein free, it won't be able to go off and fold. So when the ribosome arrives at the stop, release factor, which is not an RNA, it's in fact a protein, enters the A site of the ribosome and hydrolyzes, the ester bond connecting the last amino acid here, leucine, to its corresponding tRNA. That, as the name of release factor implies, releases the nascent polypeptide, the nascent protein, to exit the tunnel in the large subunit and go off and fold. And then the ribosome comes apart into the large and small subunit and goes off to find another messenger RNA to translate. So there's a little cartoon sketch of the relevant story. And this is the case in all cells that make proteins that have ribosomes, whether prokaryotic or eukaryotic, they all have release factors. Release factor is an essential player in this process. And as I'll explore in much more detail in part 2, ribosomes really, I would say, they are the diagnostic machine of the living state. Viruses, by definition, lack ribosomes. COVID, which is plaguing our planet at the moment, or SARS-CoV-2, it needs to hijack a ribosome from the cells that it infects. And if there are viruses on Mars, they'll be there only because there are cells on Mars that have ribosomes. So it's an essential, release factor is an essential player in this process, in all of life. It's part of the essential molecular hardware. Okay, what would have been present in the last universal common ancestor? Well, we can infer that any molecular feature ubiquitous throughout the three domains is likely to have, in other words, any cell anywhere else in the tree of life has that feature. It's likely to be present, or to have been present, that is, in LUCA. So a DNA genome, a genetic code, RNA, making, for instance, transfer RNA, messenger RNA, as I said, ribosomes, and in order for ribosomes to function, release factors, these proteins, are likely, very, very likely to have been present in LUCA. Now, they're essential, right? And that's demonstrable. If you disable the release factors or mutate the release factors in a simple bacterial cell, it's over, right? You don't have translation. Without translation, you're inviable, and there's no evolution possible. So universal common descent would predict the homology of release factors in all three domains based on what I've just described. Because if it's essential here, if it's essential hardware here, essential by definition is going to be retained as you move out into the domains and then into the branches of the tree of life. It's a strong prediction. All right, so let's do it this way. UCD predicts homologous or conserved release factors. All right, that's the setup of our thought experiment. Now, I want you to place your bet. Now, I'm talking to you via the internet, so we're not in the same classroom or not in the same seminar hall. Normally, if I would do this with a group of colleagues or a group of students, I would actually stop at this point and ask people verbally, show of hands, make a prediction. Will the release factors of the three domains be homologous or not? So if you're there listening at home or in an office or somewhere or wherever you happen to be watching this talk, do this for yourself. What's the prediction? Are those release factor proteins going to be the same? And for homologous, by the way, interpret that as exhibiting topologically in three dimensions the same three-dimensional fold. Those folds would be congruent. We could map them onto each other at the protein data bank, for instance. All right, what's the answer? Well, the answer is they're not homologous. The only thing that's shared between this eukaryotic release factor and this bacterial release factor is this GGQ motif, these three amino acids here that are the business end. These actually are the portion of the release factor that does the work of cutting that last amino acid free from its tRNA. All right, so that's the action site there. But these folds are completely different, and they were surprising when they were discovered. When the crystal structures of these proteins were solved, they turned out to be not homologous. And commentators said, well, it's doing the same thing, right? We know the role that it has to play in the ribosome, but the actual molecular tool itself, the molecular actor, is not homologous. These are not the same proteins, even though they're doing the same job. There's no homology between them. All right. Well, does this mean we need a rethink? In other words, a rethink of universal common descent, because the prediction that we just made from universal common descent went down badly, crashed and burned. Well, actually, that's not what happened. When you look at the literature of this discovery, that's not what happened. They're not homologous. That's curious, but universal common descent is still okay, right? So go into the literature, and commentators will point out the evolution of release factors holds several unsolved puzzles. There's no evidence for an evolutionary relationship between bacterial, archaeal, and eukaryotic release factors. Ergo, it is unknown how termination was mediated in the ribosome in the last common ancestor. All right. These authors in another paper say, well, you know, translation termination is an essential process, universal in being essential, but it's not universally conserved. That is, the components are not homologous, and this raises questions whether termination was essential in the last ancestral translation system. So what's happening is the anomalous data are not making their way back to challenge universal common descent. We can look at it schematically this way. There are some silent partners in this prediction business, and the theoretical partners, are taking the hit when universal common descent's predictions fail. So here is our sort of naive setup where we say universal common descent directly predicts observations. All right, you can call that naive falsificationism if you want. Because what's actually going on with the discovery of non-homologous release factors shows us the situation is actually more complicated. Universal common descent is coupled with auxiliary theories. And it's that coupling, that complex bundle here of universal common descent plus auxiliary theory that generates the prediction, right? Such that when the prediction fails, as it did in this case, the damage is paid for here by the auxiliary theory, and universal common descent is insulated. The anomalous data are, as it were, intercepted on their way back, right? A failed prediction like this tells you something over here in this bundle has got to pay for the failure. And in this case, and in many others, which we'll be discussing in more detail on the 18th, when the prediction from universal common descent goes belly up, I'm sorry, I'm mixing metaphors here. Let's stay with crash and burn. When it crashes and burns, the fault is found at the auxiliary theory. Now, most analyses of universal common descent testing would stop the thought experiment here, and they would draw their moral at this point, right? Sensible take-home message. And go back into the philosophy of science literature and say, Pierre Duhem and Quine told us all about this. When we confront our theories with the world, in other words, we've got our bundle, right? Our main theory plus all the auxiliary assumptions and theories that we're using to generate predictions, we don't bring a simple proposition up against the world for checking. We actually bring that complex bundle, and when our predictions fail and we're looking to assign responsibility for the failure, we've got to look in our complex bundle, which may be very complex indeed, and say, well, something in here is responsible for this failed prediction, logically, but it's unclear to us what it is, where the problem lies within our particular bundle. Now, and that's true. The history of science bears that out over and over, but it doesn't solve the problem for universal common descent at all. If anything, it makes it worse. Okay? Recall our original prediction. Based on the behavior of ribosomes today, we predicted that the ribosome present in the last universal common ancestor, which had a DNA genome, would have needed a release factor for the ribosome to function at all. We predicted a homologous release factors, therefore, in the descendants of Luca. Okay, now, if we take our Duhem-Quine view of auxiliary theories, we can tweak our auxiliary theory about the function of release factors and say, well, guess what? Who would have thought? But release factors can vary wildly. They may not even be functionally necessary at some early evolutionary stage. So we couple our tweaked auxiliary theory now to universal common descent, and now we can predict non-homologous release factors. Okay, this and this, non-homologous and homologous release factors, that's everything. That's all the possible data on release factors. So a prediction, well, they might be homologous or they might not be homologous. We've covered the waterfront. All possible data on release factors have been covered. What that means is universal common descent will turn out to be consistent with whatever we observe. That is not good, right? That's not good, because a theory that is consistent with whatever you can observe is not actually telling you anything about the world. It's another way of saying it's untestable. If your theory, this is a standard critique of string theory in cosmology, that it's impossible to derive a test of string theory, and it's a critique of theories elsewhere in science. A theory that is consistent with whatever you observe is not actually telling you anything. You've got to get the theory in a position where it can tell you something that might not be the case that you could discover. There's an old German proverb, if the rooster stands on the dung pile in the farmyard, if the rooster walks up on the dung pile in the farmyard, it will rain tomorrow or it won't rain tomorrow. Oh, wow. I've got everything covered. I don't know anything, right? I don't know anything about what's going to happen tomorrow because my prediction doesn't rule anything out. So this is the no answer to our opening question. Okay, if universal common descent is consistent with whatever we observe, and that appears to be the case with this first thought experiment about release factor, we have a problem. So to explore the no answer further, let's look now at a second thought experiment, the universal genetic code. Okay, now, when I was a student of biology, starting in 1980 to 1984, I studied evolutionary biology at Pittsburgh, and then from 1985 to 1997 as a PhD student at Chicago in much greater depth. In the first part of that training, the early 80s, the universality of the genetic code was a textbook prediction from universal common descent. So we've seen this slide before. Oops, excuse me. Here we have, again, features likely to be present in the molecular biology of LUCA, a DNA genome, which would require a genetic code for the passage of information from DNA to RNA via the ribosome into protein assembly. All right. And this is the abstract mapping between the realm of nucleic acid and the realm of amino acids. Here is an RNA codon table. Again, doubtless familiar to you from the biology training that you've had. I won't spend much time on this. This is the so-called universal code, which has many remarkable features, actually, that maybe we can talk about on the 18th. But again, in the early 1980s, beginning in about 1963, and then when the code was finally solved in the mid-60s, so for roughly 2 decades, as a standard prediction in the biological literature, both in textbooks but also in the primary literature, research literature, the universality of the code was taken as a strong prediction flowing from universal common descent. So Dawkins put it this way in The Blind Watchmaker: the genetic code is universal. I regard this as a near conclusive proof that all organisms are descended from LUCA, single common ancestor. And he was hardly alone in this opinion. It was the decisive confirmation of UCD. Why? Because, like conserved release factors, the centrality of the code to molecular function was seen as something that was not subject to variation, because variations in the code wouldn't affect a single protein. They would affect every protein, right? Every protein would be affected by a change in the code. So changes were thought to be inevitably lethal given their likely effect. Ergo, wherever you look in the tree of life, so the prediction held, you should see the same code wherever you move within these 3 domains. This was what I had, and pretty much everybody had as biology students in their textbooks. So we can again do it this way, right? Universal common descent predicts a universal genetic code because of its centrality to molecular function. And here's a typical expression, again from Ridley, 1985. The code would be retained because any change in it would be disastrous. No such body could live. It would change all the proteins. Thus, we expect the genetic code to be universal if all species have descended from a single ancestor. Okay, so again, place your bets. This prediction was first published in Science, as far as I've been able to determine, in 1963 in a paper in Science. We are now, what is that? How many years? Well, several decades in any case, from 1963 until today. How did it fare? Well, not very well. Here are just some of the variants in the code. In fact, there are so many variants known today that the NCBI here in the United States maintains a web page where if you are working in a group like the ciliated protozoa here, you're using Tetrahymena, let's say, as your model system, you need to go look up the variant code for your particular group. But there are variations present pretty much everywhere, and they're numerous, okay? And what's happened? Well, again, pay attention to the deeper logic. What hasn't happened is that anyone has questioned universal common descent. So here's a standard reinterpretation, if you will, of the prediction, or a reassessing of the prediction. The genetic code, initially thought to be universal and immutable, is now known to contain many variations, particular codon reassignments. All right. Now, you might look at a slide like this and you say, well, Paul, the genetic code is nearly universal, right? Yeah, there are some variants, but they're probably not very important. And you can say they're relatively minor changes. The prediction still basically holds. All right. This is a nonsense phrase. And fitting for something like Alice in Wonderland. What does this phrase mean, nearly universal, with respect to the original prediction, which was a universal code? Universal is an adjective, is not, or is a logical term, to be more precise, is not something that admits of degrees, like height or size, right? So my wife is actually taller than I am. She's 6'2", I'm 6'1", roughly. She looks down on me, literally. I mean, she loves me, obviously, but she's taller than me. Height is something that admits of degrees or size. Universal isn't like that. So if you say that it's nearly universal, what you're saying is actually nonsense, right? Because in each of these cases, you can say, well, it's still nearly universal if what you mean by that term is occupying most of some space or distribution. So 99, 90, 70, again, if nearly universal means just occupying most of some space or distribution, that could be the case, but most of X within Y is exactly not what universal means. So this is nonsense because it entails that 70 equals 90 equals 99. And when I see something like this in the argumentation of a biologist, I know he is not thinking, because the whole strength of the original prediction was its bona fide universality. Once that is lost, the modifier universal, that logical symbol, is no longer applicable. You've got to do something else. Saying nearly universal is literally nonsense. It's akin to this, or this, right? It's nonsense. This is the same problem. And unless you're writing Alice in Wonderland, avoid nonsense wherever possible. So next time, and I finished on time today. I really can't believe that. That's wonderful. Next time, on the 18th, we are going to look at the rest of the genetic code story, which is absolutely fascinating. What has happened between the elucidation of the code in the mid-1960s and today. We're going to look at ribosomes, and we're going to look at the conservation of demonstrably essential proteins in archaeal groups and bacterial groups. In other words, we're going to look at the molecular details to explore the yes answer to falsifying or testing common descent, because you can answer yes, but you have to pay attention to what it is that you're doing logically when you say, I have falsified the theory. So next time, we'll finish up with 3, the no answer. We'll disarticulate, we'll take apart these complex Duhem-Quine bundles to see where the problems are, and then we're going to look at why it matters. And I'll just preview the material on the 18th will be informed also by this ocean of surprising genetic data that didn't exist when I was a student, but do exist for the students listening to this talk, and that will continue to grow at an increasingly rapid pace within the next decade. So I really can't believe it. I finished on time.
Yeah, you did. So firstly, that was a fabulous presentation. One of the things which I was kind of thinking of is, you said almost pregnant, or, almost, you could say these things. It's almost like saying, well, there's just one part of the universe that gravity doesn't work, but it's still a law. It's just
I know. It's like that for a scientist. That's the place where you should get goosebumps.
Yeah.
Where your pulse should quicken. Treasure your exceptions. I'm sorry, Subor, for
No, I love it. I love it. I love the fact that you have these analogies, because it really breaks things down. And these complex bundles, the problem with them is that you can a priori decide, right, we're definitely not getting rid of UCD, and we've got a lot of Play-Doh. There's a lot of things here we can mess around with. The thing which is interesting about the genetic code and the supposed idea that it's supposed to be universal is because this is a point which is quite well established now. I mean, I don't think there's a doubt that this was a strong prediction that was made, decades ago. If there was some room for allowing nearly identical ideas in terms of genetic code, well, they should have been, elaborated upon before we got the disconfirmation of the data. So it doesn't really make sense that to later on say, well, it's nearly universal and so on and so forth. Well, that should have been discussed before, because these ideas are, they're out there. It's surprising that, no one had actually done that. And previously as well, we could apply the same thing when we had you speaking about orphan genes, that, well, why didn't you guys make that prediction? In fact, you made the counter prediction. So it's very clear that, the auxiliary assumptions are pretty much up for grabs, and UCD is just not going to be challenged. I wanted to ask you this question about, we are trying to, tell the public, look, there's a subtlety here which is significant, right? It's not as simple as universal genetic code, therefore this. However, what somebody could say in your presentation, Paul, is, well, it seems as though this presentation is touching upon the science, but it's really more philosophical. And the problem here is people like Richard Dawkins, they don't like the philosophy of science. They don't like indulging in it. In fact, at the World Atheist Convention in 2011, when someone asked him about the philosophy of science, he said, just do the science, don't worry about the philosophy, right? In other words, so if we're going to have that attitude, then in a way, couldn't one say, well, Paul, your arguments are going to go unheard because people are not really accepting the intersection between science and philosophy?
No, and that's a legitimate concern if I wasn't going to do the second part.
Right.
Philosophy of science that does not engage as fully as possible with the evidence is sterile. It ends up being sterile. But I don't put a bright line between philosophy on the one hand and science on the other. I think one passes between the two realms. You hardly need a passport, right? You can just walk right over there. Or if you're in science, come back over into philosophy. And good scientists do this all the time.
Yeah.
They're constantly saying, well, why am I holding this assumption? Is it because I've actually established it? Or is it just a convenient premise that I've adopted for whatever reason, but actually doesn't obtain?
Yeah.
So the reason that I started today with the logic is so that when we do engage the data in detail in 2 weeks, I won't have to go back and say, and oh, by the way, notice what's happening here. Universal common descent is being insulated, and the auxiliary theory is paying for all the damage that was caused, right? So really, it's important, I find, to do a certain amount of ground clearing, a certain amount of logical analysis before you go into the data, so that you understand the significance of the data when you're grappling with them. Yeah. But I personally would never be satisfied to leave the talk where I ended today. But I mean, I can't go for 3 hours, right? Yeah. Yeah. And when I'm actually, frankly, a little concerned about the length of the material for the 18th, because I'm going to finish the genetic code story, look at the ribosome, which I've learned things about the ribosome over the past decade that just, not personally, experimentally, but from absorbing the work that others have done and thinking about its implications. I hope to convey that excitement to the audience next time. But frankly, the patterns of anomalies that we'll be looking at extend broadly over biology.
Yeah.
And it's really been whole genome sequencing that's enabled us to see this in a way that really, if you had told me even in the mid-90s as preparing to defend my dissertation, I simply wouldn't have believed it.
Yeah.
So I don't like logic chopping.
Yeah.
If it's and conceptual analysis, if you leave the story there, you've got to go into the evidence. And so I'm putting out a big ad for next time.
Yeah, sure. I was going to say what we could do is perhaps split the presentation into 2. So we have one on the 18th and that another time that you're free. Because I'm going to make this into a playlist on YouTube so people can watch over time. I was also just going to add something. There was a part, I think near the beginning, there's a part I was remembering, when Philip Johnson spoke about naturalism and, in his book, The Wedge of Truth, essentially that's what it's really going to be. Essentially, that's the real issue here, because if we go with this idea that life only comes from life and life only arose once in the distant past, then UCD is just inescapable from a logical point of view. And if it isn't inescapable, then continuously, putting it in that complex bundle, it just completely insulates it it's just bulletproof, right? You just can't do anything about it. So I think on the minimum, why this particular presentation and these sort of arguments are very important is because at least we can clarify to the Darwinists that, well, we have tried and this is the result, and clearly the data is showing something different to what was predicted. Now that may not lead anywhere, it may not challenge UCD in biology, but what it does do is it does leave them gobsmacked because they don't actually have an answer to this. I was also thinking, Paul, that you were you've been around in that transition period, and perhaps if you studied biology later or in this period, you wouldn't have been a witness to these things that you experienced. These aren't just historical things, these are things that you experienced as you're going through academia at that period.
Yes, I think that's especially important, that when you live through an episode like this. I remember, in fact, when the first variants in the code were discovered. They were known in ribosomes from the late 70s, but because the protein census of a ribosome is relatively small compared to a whole organism, people said, well, a ribosome can probably handle some variation. Then the first genuine variants in the nuclear code were discovered, and then they just kept coming one after another. So the NCBI does actually an annual update of their page where they list the variants. I do want to point out that it's possible to jettison UCD and stay within a naturalistic framework. So next time I'll talk about, for instance, W4 Doolittle and other evolutionary biologists who, for at least 2 decades, have said, I don't accept that single tree picture, yet they remain naturalistic evolutionists. Actually, in the 19th century, Darwin was very much the exception in arguing for a single common ancestor. Ernst Haeckel, the German embryologist, thought that spontaneous generation was happening all the time.
Yeah.
So that the Monera, the single-celled organisms that were you could see in a microscope today were not the lineal descendants of Monera from early on Earth history. Jean-Baptiste de Lamarck had a polyphyletic view. So the conceptual landscape has at least 4 major quadrants, one of which is design and universal common descent. Mike Behe has a view like that. Michael Denton as well.
Yeah.
And I try to encourage students not to look for a simple polarity where it's either miraculous special creation or naturalistic evolution. In fact, it's a complicated landscape.
Absolutely. And I would say this is a very exciting time for people who are getting involved in this field, because in a way, the conceptual framework is laid before you. It's very clear what needs to be researched. And I would say, in the 90s, we were quite disadvantaged. We didn't have these things. All we could really do is very similar to what Philip Johnson was doing, was simply point out the elephant in the room, but now we can actually conceptually break down the elephant and point out the various problems with it. So Paul, I'm really grateful for your time. Next we're going to be on the 18th, and then we'll see when you're free after that to do the following episode. In terms of you guys who are watching this live, please share, like, and follow Paul at the Discovery Institute. Paul, is there any publication or writing that you've recently been involved in?
Actually, I have a chapter on this very topic coming out next month in a multi-author anthology, and I will send you the information about that if you wanted to make it available in the notes for the YouTube, the YouTube entry. I'm actually working on a book about this topic, looking not just at the base of the tree of life, but up into the branches as well, where we see the same pattern of protecting common descent and making the auxiliary theory pay. And I'm mostly spending time these days on orphan genes.
Yeah.
So we have an international research project called Orphan Base, and one of our members won a Fulbright to come to the States and write code to detect orphan genes. So it's a very exciting time. Biology is the most exciting science to be in right now, I think.
Absolutely. So thank you, Paul, for your time. For everybody else watching, until next time, Assalamu alaikum.